name is Amit, and I head the R&D of MedGenome. So we have two speakers who are, who are into this cancer vaccine neoepitope area. They're both coming from East Coast. But before we go into the speakers, I just wanted to give you a little background on why we are interested in, neoepitopes and some of the things that we do at MedGenome. So we are a genomics company. As you can see, that our lab is on the other side of the building, and it's a NGS lab. We have next generation sequencing machines, and what I wanted to show you in a couple of slides is how we are trying to apply genomics in the area of cancer immunotherapy. And so this is the excitement that is taken the maybe the world by a little bit of unexpected surprise. So here we have the tumor cell which needs to be killed, and here is the killer cell which is the CD8 Tcell, and this is the cell that activates the T cell so that it can go and kill the tumor cell. And this activation is mediated by this complex, which is the peptide MHC complex presented by the dendritic cell, which is the antigen presenting cell, activating the T cell through the T cell receptor, and then the T cell goes into the tumor or in the tumor microenvironment, seeks out a tumor cell that is presenting the same peptide on the MHC molecule, and this recognition will kill the tumor cell. That's the point, right? Now, there are many molecules. Now there are 3 molecules which have been approved, and these molecules block the interaction between these 2 proteins, and these are the proteins that keep T cells in check. I mean, they have evolved during our evolution just to keep the T cells within control, but the tumor has co-opted some of these function by expressing these molecules on their own cell surface, and that keeps the T cell from killing the tumor. So by using these antibodies that will block this interaction, the T cell is there to kill the tumor cell. So that's the premise. So where is the genomics coming into this picture? So this is summarized in this slide. As you can see, that the peptide which is presented by class 1 MHC is derived from mutations that are found in the tumor cell, because tumor cancer is a genetic disease. Tumor cells accumulate a very large number of mutations, and the peptides that are generated from these mutated proteins can sometimes activate T cells. So identifying these peptides from the mutations is one of the areas that is very hot in this cancer immunotherapy field. And tumor mutational burden comes from exome sequencing, which is the application of NGS in this area. The second aspect is the tumor microenvironment, because the tumor microenvironment plays a very important role in regulating whether the T cell would kill the tumor cell or will be suppressed. And there are many components of the tumor microenvironment that one can analyze by from the RNA sequencing data. And I'll show you that we have built some tools to analyze T cell neoepitopes, to analyze the tumor microenvironment. And all this together can actually give you biomarkers that can predict response to immuno-oncology drugs. So this is our platform. It's a end-to-end genomics platform. We call it OncoPept because it is the peptide which is very key in mediating interactions between T cells and tumor cells, or T cells and dendritic cells. So we have built the OncoPept TUME, which is the tumor microenvironment analysis, and OncoPept VAC, which is the cancer vaccine. And this is the cancer vaccine or neoepitope would be the basis of today's presentation, and we have both speakers, and I'll introduce them after I finish my talk. And then there are other solutions that we have built in terms of TCR sequencing, in terms of predicting toxicity. So this is the interactions between the tumor cell and the T cell receptor, And these are, for example, as I mentioned, that somatic alterations can generate peptides that can bind a T cell receptor. And this T cell neoepitopes, if one can identify it, they can improve the patient's response. They can be used as cancer vaccines. So there are a lot of applications of these peptides. So this is a standard pipeline that everyone uses to prioritize neoepitopes. So you you take the tumor tissue, you do DNA RNA sequencing, you identify all the variants, you look at their expression, and then you pass them through HLA binding, which is the binding of the MHC class 1 molecule to the peptide. And then finally from this you can identify the neoepitopes or potential neoepitopes. But we found that this particular pipeline has certain limitations, and this is shown here. So the red line is our peptides which are known to be immunogenic. So these peptides can activate T cells. But if you look at the red line, this is the region which are all false negative. So which means that these peptides in the standard pipeline will be excluded because they have a affinity of binding which is higher than 500 nanomolar, which means it's a weak binding of the peptide, and they will be excluded from your analysis. So you're actually losing a lot of peptides in this region. Then look at the blue line. So the blue line are peptides which are non-immunogenic. So they are not going to activate T cells. But as you see that you are going to select these peptides which are false positive because they have a very high binding for HLA less than 500 nanomolar. So if you are dependent on HLA binding alone, then you are going to select a lot of peptides which are false positive and exclude peptides which are false negative. So we thought that there is one area that still missing is how are these peptides interacting with the TCR. So we built this TCR binding algorithm, and I won't go into the detail. So we built this algorithm, and I'm just showing that if you use the TCR binding algorithm versus HLA binding and just compare what is your sensitivity, specificity and accuracy, you can see that you get a much better accuracy, sensitivity and specificity just by including the TCR binding algorithm. So this is something that is unique to our pipeline, and so we use TCR binding as one of our criteria for selecting peptides, and of course we put HLA binding, TAP binding, all the rest of the elements which are there in the standard pipeline. But by including TCR binding, which is this step where the peptide is presented by MHC class 1, and then the T cell receptor or the T cells are floating around screening the peptides for a binding partner, and not all T cells will bind to this peptide MHC complex. It's one or a few T cells which will do. So we predict this interaction, and we find that by predicting this interaction you increase the accuracy of prediction. So we are trying to work with, for example, Professor Srivastava to validate our pipeline. We're working with Beatrix on mass spec. So they will discuss that. The second thing that we built was we built the tumor microenvironment analysis solution by creating gene expression signatures. I won't go into the detail, but we have taken a lot of pain to create the gene expression signatures. We validated, and I'll show you in the next slide. So we have analyzed a very large number of microarray data set, RNA-seq data set in order to arrive at our signature. And as you can see that the signatures, I'm just showing you the 3 signatures that we have built for CD8, CD4 and Treg, and these are very important. You can see that it distinguishes this cells from some of the other cells, both here. This is from microarray data, this is from RNA-seq data. So how are we using this? So we, as I told you that RNA sequencing of the tumor will give you transcripts, whole transcriptome analysis, and we apply our gene expression signatures in order to immune phenotype the tumor. And the goal is to understand the tumor in much detail in terms of their cellular content, in terms of their network, cytokine and chemokine network. This is a graph showing the CD8 Tcell infiltration in all the 33 cancers in TCGA data set. These cancers have been divided according to their mutation burden. And as you can see that the mutation burden is not correlated with CD8 T-cell infiltration. So, high mutation burden does not mean that it will have CD8 T-cell infiltration, but one thing we know from clinical trial is high mutation burden is a positive predictor of response to cancer immunotherapy drugs. So, what is happening here? So, what we found was that although the mutation burden does not predict response, if you calculate how many T-cell neoepitopes are there in these tumors, the T-cell neoepitope burden correlates very well with the CD8 T-cell infiltration. So, we know that CD8 T-cell infiltration is critical for cancer immunotherapy drugs to work. So, there is a correlation between CD8 T-cell infiltration and the T-cell neoepitope burden, and that might explain why mutation burden correlates with response, but mutation burden may not correlate with the CD8 T-cell infiltration, the amount of CD8 T-cell infiltration. This is just to show that if you separate tumors that are high in CD8 T cells, and these are 3 different tumors. Melanoma is highly immunogenic, head and neck cancer is somewhere in the middle, and prostate cancer is very highly non-immunogenic. And you can see that the CD8 T cell content is only 1% in prostate cancer versus about 30% in melanoma. But what is quite remarkable is that in these 3 different cancers, and we have looked into other cancers, there are some common pathways that are associated with CD8 T cell infiltration, such as pathways related to antigen presentation, PD-1 signaling, and so on. So, CD8 T cell infiltrated tumors have some common properties or common characteristics. This is showing that the CD8 T cell tumors that are rich in CD8 T cells are actually poor in CD4 T cells. So, there is a inverse relationship between high CD8 and low CD4. So, like prostate cancer is full of CD4 T cells, and I showed you earlier that it is full of it has very less CD8 T cells. And also the interesting thing is that when you look at this CD4 high tumors, they define a completely different set of pathways that are enriched in this tumor. So, there is something interesting biology going on between the tumors based on the tumor microenvironment. And this is my last slide, that we use OncoPepTum for a variety of clinical utilities, and one is to actually identify tumors that might respond to cancer immunotherapy drug, and based on the CD8 T cell infiltration score, based on the interferon gamma score, you can actually group them into these 3 groups, and you can make a prediction that one will just respond to checkpoint blockade, the other will not respond to checkpoint blockade, but may benefit from other therapies, radiation and others, to kill the tumor cells and produce more neoantigens so that they can bring in CD8 T cells. And there are a few other areas that we are currently working on to so this is a summary that our goal is to apply genomics into in this area of cancer immunotherapy. We know that there are tumor intrinsic factors defined by some of these elements which are important for checkpoint or response to checkpoint blockade. There are tumor extrinsic factors which are defined here, which also regulate how the checkpoint molecule will work on tumors, and 2 together can give you a much better predictive marker of response. So, our goal is to work with academic institutions, even companies we are working with, and giving them as a service. So, this is the conclusion, and so I would you can ask me questions maybe later on.